All right, good evening or good morning, depending on where you're joining us from. It's another Wednesday. We get to experience and share knowledge with each other as regards soil science. And today we get to hear from Dr. John Kovar. But first, I would like to do the land acknowledgement. Iowa State University aspires to be the best land grant universities at creating a welcoming and inclusive environment where diverse individuals can succeed and thrive. As a land grant institution, we are committed to the caretaking of this land and would like to begin this event by acknowledging those who have previously taken care of the land on which we gather. Before this site became Iowa State University, it was the ancestral lands and territory of the Bakoje or Iowa Nation. The United States obtained this land from the Meskwaki and Sauk nations in the Treaty of 1842. We wish to recognize our obligations to this land and to the people who took care of it, as well as the 17,000 native people who live in Iowa today. Today we get to hear from Dr. John Kovar, but who is Dr. John Kovar? Dr. John Kovar is a research soil scientist with the USDA-ARS National Laboratory for Agriculture and the Environment and an affiliate associate professor of agronomy. He received a BS in agricultural science from the University of Illinois in 1981 and a master's and PhD degrees in soil fertility, plant nutrition from Purdue University in 1985 and 1989, respectively. Before joining the staff at NLAE, he held a faculty appointment in the Department of Agronomy at Louisiana State University between 1989 and 1999. Dr. Kovar has more than 40 years of professional experience, ranging from industry to university and government service. He's recognized for expertise in nutrient management, soil-plant relationships, soil testing methodology, and water quality issues. He's the author and co-author of more than 130 scientific manuscripts and publications, and has served as an associate editor of the Agronomy Journal and Journal of Environmental Quality, as well as a contributing editor of the Fluid Journal. He's an active member of the American Society of Agronomy, Soil Science Society of America, and the Soil and Water Conservation Society. At this juncture, I would like to invite Dr. John Kovar to give us and share with us some of his knowledge.
All right, thank you for that introduction. And thank you for inviting me to present this seminar on some of our work. As you can see by the title, the LTAR Common Experiment is what I'll report on today. So the first thing I want to do is ask who here knows what the LTAR network is or anything about it. You familiar with it? Okay, a couple of people and that's it. So, all right, so I'll talk about that here in a minute in much greater detail. So today's story, what is the LTAR network? Since most people don't know, what is the common experiment? That was also in our title here. So what is that and what role do the folks at the NLAE play in this common experiment? What have we discovered? I'll throw out a few things here that we found out since we've been doing this. And then what does the future hold? So what do we plan to do coming up? So here's a map. Wow, that looks different on the screen, but that's okay. Here's a map of the sites. So the Long-Term Agroecosystem Research Network, that's LTAR, the LTAR network encompasses about 18 different sites around the US. And they're shown on this map here. I don't have a pointer, I guess. Will this work? Yeah, this should work. I think that shows up. So yeah, throughout the US, it was created in 2011 or thereabouts. Mark Walbridge and Steve Schaefer in 2011 published a paper on their thoughts about forming a network. This was in response to a lot of questions about what was going on at ARS benchmark watersheds around the country. There were 28 or 29 of these that were collecting long-term data. Some of them had been in operation for more than 100 years at that point. So they wanted to somehow coordinate these different sites. And so they wanted to set up this network to do that. They modeled it after the National Science Foundation LTAR Network, the Long-Term Ecological Research Network, which was established in 1980. So this was set up specifically to look at ag-related problems. Later on, about 2014 or so, the concept of sustainable intensification came into play. Sherry Spiegel and several other folks published a paper in 2018 relating this sustainable intensification idea and how this concept could be used within the LTAR network. One thing they thought about was you need local effort. So we can't compare things. For example, they don't grow sugarcane in North Dakota, so there's no reason to try to compare some system like that. So it has to be more of a local effort. And it advanced the concepts of business as usual systems. So what's being done in that local area now versus some aspirational system. So how can we intensify agricultural production in a sustainable manner? That was the idea here. So after that, 21 independent but coordinated experiments were established at that time. This was about 2016, and that's when I got involved with the LTAR network. It's about 2016. So here in the upper Mississippi River Basin is the site where we're working. What were the problems here? What are we dealing with here? So intensive agricultural production here means corn and soybean production. That's what we grow around here. And you can see the states, Illinois, Iowa, we trade places there for top producer in the country. Minnesota's involved. Indiana, Nebraska. So we all produce a lot of corn and soybean in the upper Midwest here. So that's our intensive production. But what about sustainability? A major sustainability issue that we have is nutrient export. And you can see here, will that be on your right? On the top, we have nitrogen yields from this area. And you can see the darker colors show a lot more nitrogen. The darker the color, the more nitrogen that we lose. The bottom is phosphorus. So it's the same thing. Phosphorus export, we lose a lot of phosphorus as well. So we're losing these nutrients with this intensive agricultural production. So that's certainly a sustainability issue. Another issue would be soil erosion and soil health. So we have to protect our soils. We're losing a lot of soil to erosion, certainly in hillier areas of the landscape around this upper Midwest. Not so much where we're working specifically, and I'll talk about that in a minute, but we are losing soil. So soil erosion is a problem. And you can see on the map over there on the left that the darker areas mean more soil erosion. So we don't have as much of a problem in central Iowa, but there are other places where they have more. So the darker areas where hill slope erosion is a big problem. So for our site, we are in the upper Mississippi River Basin site, as I alluded to earlier. So within this site, we have 4 locations. Ames, of course, which I'm going to talk about today, but also in Morris, Minnesota, there's an ARS location there where they have a common experiment as well. There's a location just south of St. Paul, the ARS location as well, that they have a common croplands experiment. And then there's a location that's run by the University of Wisconsin at Platteville. And they also have a common experiment. However, the Platteville location has a grazing lands common experiment because they do a lot of grazing over there. And so they're interested in sediment and nutrient losses and erosion over there at the Platteville site. But they are helping with the Ames site as well. So we have these 3 sites that are dealing with croplands, and so corn and soybean production. And let's see, I have some photos here too. So the Morris site, there's one, their reduced tillage experiment. So they're looking heavily at tillage. St. Paul's looking at cover crops as well as the Ames location, our location here. We're interested in cover crops. And again, at University of Wisconsin-Platteville, they're doing more grazing work there, but they are helping with our croplands as well. Who contributes? I threw this slide together and I hopefully have gotten everybody on here that's contributing from the lab. So we have research scientists, several of us that are working on this and have been for a while. We have retired scientists. So again, this started in 2016. A lot of the people that you see here, retired scientists, were involved in 2016. Even before I was involved, they were involved with it. At that time, Tom Mormon was actually the Upper Mississippi River Basin site lead. And when he retired last year, he left that job to me. So now I've started doing that since that time. We also have tech support folks that help out and really make this thing run. We have a data management team, Kevin Cole, Michelle Kreider, and Samantha Purdy, that help collect all the data and organize all the data. Because as you can imagine, coming from these sites, you need a data manager to take care of all the data that come in from the various scientists that are working on it. So we have to centralize that and make sure that we capture all the data and everybody knows where it goes. So that's very important. We have a lot of tech support here from the agronomy and soil science side, keeping things running out in the field, collecting the samples, helping collect the samples. And then I didn't list here, but we have a number of student workers that we've hired that work in the lab as well and go out and collect samples with us. So it's sort of a team effort at our lab then to get all these things done. When something needs to be done in the field, we take a small army of people out there and get it done quickly and get all the samples or the measurements or whatever we need. And it seems to work pretty well. The analytical lab, we run all of the water samples through that lab. As you'll see, I've got some data on that. And some of the soil samples, and I think in the future we'll run some plant tissue samples through there as well. At the Upper Mississippi River Basin site in St. Paul, Dr. John Baker is the lead there. He's an ARS scientist as well, and he's got a whole crew the same way we do, data management and other tech support folks working with him. At the Morris site, Dr. Jane Johnson, she's also a research leader there and runs their site, and they also have people there. Dennis Bush, Dr. Dennis Bush from the University of Wisconsin-Platteville is the site lead at that particular location. And so he also has other folks working with him as well. So again, this is quite an effort. The Midwest Climate Hub, we've gotten involved with them recently. So Dennis Toddy and Lori Nowatzki now are working with us from the Climate Hub to help figure out what all the climate change information is going to mean and how that's going to affect production. So now basically, I want to look at just our site now and give some information about that. So our common experiment is at the Iowa State University Kelly Research Farm, northwest of town. There are 24 plots there. Each of them has an individual subsurface drain. So we can capture the tile drain water, the subsurface drainage water from each individual plot that's out there. These were established in 2001, so they're more than 20 years ago that they've been out there. Of course, it wasn't part of the LTAR experiment until 2016. There are 6 treatments out there and 4 replications of these plots. And corn is grown in even years and soybean in odd years. And so the aerial shot there shows the entire location. And then I've got just a photo here showing the individual plots that are out there. The plots are 40 rows wide, 40 30-inch rows of corn or soybean are grown out there. So then I've got a table here that I put together showing the protocols for each of the field experiments. First of all, we have our basic practice. There's a photo of that. The basic practice is just what they do around here in central Iowa. And that would be a fall chisel plow and a spring disc before corn planting. We put our nitrogen fertilizer down as anhydrous ammonia at a set rate, 166 kilograms of N per hectare. There is no cover crop there. So this is just a basic practice that people would use in this area. Then we have a no-till system. Very similar. Only we use 4R nitrogen management in those particular plots, and there is no cover crop. So what do I mean by 4R? Well, if you look down at the bottom here, I added some information. So we have a fixed starter nitrogen rate that we put down, 33 kilograms of N per hectare. And then we come back later on at about the V6 stage. So when the plants are less than a foot tall, we come back and do a side dress application. That's based on results of the late spring nitrogen tests that we run out there. So we go out and soil sample, figure out what nitrogen's in the soil, and then come back and apply urea ammonium nitrate and inject that into the soil on these plots. So that's our precision management there. So the 4 R's again are right rate, right time, right place, and right source. So that's what's different than the basic practice. We also have a no-till system where we use a rye cover crop. That's shown in the photo here. And again, that's 4R nitrogen management. And the cover crop is dropped into the standing crop. So it's planted while the crop is still out there. And it follows both the corn and the soybean phases. And then finally, we have our relay crop. And I'm going to explain this a little bit more in the next slide. But for the most part, I can explain a little bit here. So we disc the field after corn is planted. So the corn residues, we disc those and we plant the Camellina into that. After soybean, it's a no-till system. For fertilizer, we use 4RN management, but the Camellina does get about 40 kilograms of nitrogen per hectare to help it grow. And again, our idea here with this relay crop is to have a crop of Camellina that we can harvest. So we're trying to actually grow a crop there. The Camellina, let's see, is after the corn harvest, so we plant that. There's an oat, radish, field pea cover crop mix that's grown after soybean. That may change in the future, but at this point, that's what we've done. And again, we use a shorter season corn hybrid to grow in this relay cropping system, because we have to harvest the corn, then disc the field, then plant Camellina. So that's why we use a little bit different system here. So the Camellina then. Here we go. So I guess I ran ahead a little bit here, but that's okay. So the Camellina, we plant that in October after the corn harvest. It overwinters and then it grows into the spring, and we harvest it in late June or so. So you can see a shot here. I don't know if you can see this very well, but there are some small Camellina plants in here that there's a row of them that's hard to see there, but they are growing in late October. So they emerge and start growing. Then they overwinter in late April. This is what it looks like. Maybe not this year, but we're not growing any this year. But again, this is what the crop looks like. If you look real hard, you can see there are some skips in the Camellina. So we have skip rows in there. This is where we come back and plant soybean. So the soybeans planted right into the growing Camellina crop. If you look right here, you can see our soybean plants are emerging here as the Camellina is starting to flower. So this is where our relay comes in. So we actually have 2 crops growing at the same time on that piece of land, or in those plots. So we have both the Camellina and the soybean growing. So the Camellina gets harvested in mid to late June. We come in with a combine and harvest right over the top of the growing soybean. So the Camellina is tall enough. The seed pods, this is an oil seed crop. It has pods, siliques, I believe they're called. And you just harvest that plant or that crop right over the top of the soybean plants. And then the soybeans are allowed to grow. So that's where our relay system comes in. So we're growing corn, Camellina, and soybean in the same land area in 2 years instead of 3 years. So what's aspirational about this, all these various treatments that we have? First of all, for our nitrogen management, we're changing that up. It's more difficult. There's more management involved when you do these split applications. It's easier just to go in with anhydrous either in the fall or spring or whenever you're going to do it and get it on the crop and you're done with it. So there is more nitrogen management involved with this 4R approach. Cover crops as well. So we think they're still aspirational. I wish they weren't, but they are. So the Iowa nutrient reduction strategy had a goal of about 12.5 million acres of cover crops growing in the state. The last number I remember seeing was about 2.2 or 2.3 million in 2020 maybe. So it's probably a little bit higher now, but still it's a far cry from the 12.5 million acre goal. There are barriers to adoption. Of course, there's a cost to it. There is some support from IDOLs and NRCS, depending on what you're growing, if it's your first time or second time or what. So there is some support for this. And the range is about $9 to greater than $35 an acre for this production. But it still costs you more, from what I recall, it costs a little bit more than that to actually grow a cover crop. And so you're not making any money on this by any means. There's the timing thing. It's another operation to consider. You have to get the cover crops in. So you're trying to get your corn crop out or your soybean crop out, and then you have to figure out how to get the cover crop in. There's some thought that yield drag is still a problem. I think it is. There is some indications that, yeah, we need to do more work on that. There's more research to figure out how to manage these crops. So the rye cover crop will decrease the corn yields, and we can't have that if they're not making any money on the cover crop. So can Camellina solve that problem? Is there yield drag from that? Can we produce enough of the oil seed to offset the cost of actually producing the crop? And can it act as a true cover crop and capture some of the nitrogen the same as the cereal rye would do? We didn't know that. So that's one of the reasons to set this up. And then at this point, I have to say, Camellina, the adoption of that is zero. It's nil. There's nothing going on. There's a lot of interest in it, but nothing going on yet. I know there's a similar crop as pennycress, and there's a company in St. Louis that I saw advertising. They called it covercress. So their own variety of this crop that could be grown for oilseed, the same way as this Camellina. And there's a lot of interest in Minneapolis. In Minnesota, the group up there at Morris has been doing this longer than we have. And I think they have their system down a little bit better, at least for that area in Minnesota. I'm not going to go through all these, but these are all this is all the information on data collection, what we collect in the field. So if you read through this real quick, you can see we collect a lot of data, all the crop production data, the management data. We're looking also at not just the grain production, but also what's in the grain, what's coming off the field. We're measuring nitrate, total and dissolved phosphorus, total potassium, and total sulfur in the drainage water that comes out of each of those individual lines. We're looking at nitrous oxide N2O emissions at weekly intervals. We also do several soil measurements here that I won't talk about today, but a lot of these are for soil health purposes. So we're interested in soil health in these plots, and we've been sampling for quite some time to collect those numbers. And then, of course, weather data. So we collect all the weather data of what's going on out there over time. So now I'll talk a little bit more about the field itself and show some data, some results, I guess, if you want to call them that, what we've learned. The soil out there is by and large a Canisteo silty clay loam. So here's a soil map showing that. You can see the number 507 here is the Canisteo. It's probably 70% of that field is Canisteo soil. There's a band of Nicolette loam going through the middle here. And finally, there's a small area, a couple of small areas on the southern end of the field or field site there that's Clarion. So by and large, low slopes. So we don't really address runoff, surface runoff from these plots because there really isn't any. So we don't have to worry about the erosion and losses with surface runoff. Soil properties then. So I pulled samples. I've pulled samples every year, but these were a set of samples pulled from the entire area in 2016 when we were starting the project in the fall. And so you can see the organic matter, pH, the Bray, phosphorus, exchangeable K, calcium, magnesium, and extractable sulfur in the field. These are the things that I was interested in from a nutrient management standpoint. And so you can see our ranges as well. I will say for the Bray P values, for the most part, all these plots are well above the optimum level of 16 milligrams per kilogram. So yeah, so we don't really have much of a problem with phosphorus out there. There was phosphorus put down in the fall of 2016 before I had all these numbers to see what we had there. Potassium, again, I probably should have added the median instead of just the mean because the potassium is more of a problem out there. So there are a lot of plots that are below this 161 part per million optimum out there. So we have been adding K quite a bit, but we haven't added much phosphorus at all. There's a little bit that goes into a starter fertilizer. Sulfur, I don't know. Again, folks use 10 milligrams per kilogram as a critical value, but I don't know what that means, if that's true or not. So ours was 7, and we had quite a range there. So Camellina, like canola and some of the other mustards, require sulfur to grow well. So we have been putting sulfur down on those plots. All right, so I have some corn production, some soybean production data, and some Camelina production data, and we'll look at how things are faring out there. So this is, again, corn is growing even years. So I have 2016, 18, and 20 up here. I want to point out a few things here. So on the x-axis here on the bottom, we have our basic practice, our relay cropping system with the Camelina, just the no-till system, and then the no-till system with a rye cover crop. So those are what's shown here. I guess, so we have a cover crop versus a no cover crop. We also have the 3 systems have basically 4R nitrogen management versus the basic practice. So that's what we can compare here. So if you look at 2016 in our corn yields, we had some yield drag here. Our no-till system was much higher, significantly higher than our no-till system with a rye cover crop. So we had some problems there. We didn't see that later, but we had lower yields than the basic systems, the basic practice system as well. So we didn't, yeah, but we saw no difference here. So the in this case, we didn't do as well with the 4R in management as we should have, because it's lower than the basic practice. Soybean production. So this is from 17, 19, and 21. So these are the yield data from that. You can see every one of those years, well, I guess 2019, there was not a significant difference among the yields, but in 17 and 21, we got significantly lower yields in the soybean plots where we were growing them as a relay crop versus as just a regular crop. And so we lost some yield because those plants were slower growing because they were shaded and possibly for other reasons, but we lost some yield in that relay system. Camelina production, I think this is a bit low compared to what Minnesota and some of the other places that are growing Camelina are growing. I think we're still in a learning curve with that. I think we had, Kent, you could probably vouch for that, that we've had some harvest losses of Camelina seed because these seeds are so small that it's hard to capture those. So yeah, that might be a problem, but we have a new combine this year. And so the next crop we plant, hopefully we'll do better there. 3-year totals then, if you look at these, you can see that for corn, the basic practice, we still have a lot of work to do with our management to figure out how to grow corn versus the basic practice, still the highest yielding practice. For soybean, no, not so much. You see the relay crop there is less, but soybean did well. So our other systems did well. Of course, we're not managing nitrogen in those systems, so you'd expect it to do well. The Camelina, yeah, that's average again, 3 years of production. Now I will say, if you look at the soybean production and the Camelina production, so if you consider that as one unit, since we're growing it as a relay crop, then if you add those two grain production or grain and oilseed production together, economically, maybe we are doing okay compared to the other systems. So I don't know, we haven't run any economics on this yet. So now some other issues. So the sustainability part. So water quality and air quality, I've got some numbers on that. So this is for water quality. Again, we're collecting tile drainage water, looking at nitrate, ammonium, phosphorus, and potassium and sulfur. And I won't present data on all these things. We have N2O emissions being collected. We have chambers out there that are sampled at least once a week. In the middle there, I have a slide here, a photograph. These are new continuous sampling chambers, as I understand it. So Brian Emmett put those out earlier this year. So we should have continuous sampling, and that will be better than the weekly sampling that we're doing right now. Get better data. So nitrate losses in drainage water. So here, nitrate load, here are on the bottom, on the x-axis, we have our 4 practices here. So we've lost in this situation, we've lost more. There's a lot of variability, but we lost a bit more with the no-till system without any cover crop. What I want to point out, though, is how much lower planting that rye cover crop got our concentration, our loads of nitrate that we lost. Tremendous drop, like more than 50% loss, and decrease in the losses and by planting that rye cover crop. The aspirational system, we lost a lot. So did the Camellina not work? Did it not capture any nitrogen like the rye did, the cereal rye did? Well, here's a problem we have. And Brian Emmett figured this out looking at all the data over time. What happens is in the fall, as I mentioned, we come in and do some tillage, some disking of the corn residues, and then we put a little bit of ammonium sulfate fertilizer on. When you add that fertilizer with essentially a planted crop of Camellina, it's not capturing anything. And so in the fall, we're losing some of that nitrate in the tile drainage water. So there's high post-corn harvest nitrate concentrations that are coming out in that system. So then the thought is, well, maybe we don't need the sulfur or the nitrogen in the fall if we're going to capture the nitrate that's coming out with that crop. I don't know if we need that or not. Since we're still learning, will we lose Camellina yield if we do that? We don't know. But maybe we should look at it more from the environmental quality standpoint and not put any nitrogen or sulfur down there. For N2O emissions, then, you can see that for our basic practice and the relay crop, again, we're losing more N2O from those two systems compared to the no-till systems out there. So in this case, it would imply that the 4R nitrogen management is helping with the N2O losses, and we're losing less nitrous oxide. Switching gears, this is phosphorus. So part of the LTAR network, there are more than 20 groups in the network. Part of that, there's a group working on phosphorus budgets at a lot of different sites throughout the country. So we were involved in that. I've got the red circle there showing the particular system data that we included here. But you can see if you look along the top, there are systems that are grown for biomass. There are cropping systems. There's a mix of the two. There's pasture systems, and there are range systems here as well. So this shows the phosphorus balance in the system. So above and below, if you're losing phosphorus from the system, you're below the zero line here, the horizontal line. If your system is gaining, then you're above, and so that would be the green bars here. We submitted only 2 years of data from our plots here for the business as usual or basic practice system, the no-till, and a no-till plus rye cover system. The budget for these three systems over these years was nearly balanced. So the losses equals the losses and removals equal the inputs. So there wasn't much input in the first couple of years. One application of fertilizer, and then about 9 kilograms per hectare, I think, for the starter fertilizer application. So there wasn't much. The losses in the subsurface drainage for our plots were about 0.3 kilograms of phosphorus per hectare per year. So those numbers are really small compared to how much was exported into grain. So the corn grain exported about 33.5 kilograms of P per hectare, and the soybean about 20.1. So the 0.3 kilograms is very little. What effect that has on water quality, that's a different beast there. So we'll have to monitor that, because obviously from a farming standpoint, 0.3 kilograms per hectare per year is nothing. But maybe from a water quality standpoint, it will have an effect. So what have we learned here? Good, I'm gonna have some time for discussion and questions here. So after 6 years, our corn yields were highest under the basic practice. So we still have some things to do to figure out how to get the other systems to yield as much as the basic practice. Soybean yields were lower in the relay cropping system, but that could be because of light competition, maybe even water competition. We haven't explored that as yet, but we tend to look at that. So are the plots with the relay system losing less water over time than some of the other plots as far as the amount of drainage coming out? I don't know that. So, but for some reason, the soybean yields were a little bit lower. The cereal rye cover crop is effective at reducing N loss to the tile drainage by about 57% here, but it's less effective at reducing N2O emissions. The 4R nutrient management was effective in reducing N2O emissions, but not N loss in the tile drainage. So we didn't do as well with tile drainage losses. So there are contrasting controls on N loss that we must consider. So one management practice may be good for nitrous oxide emissions, but you still lose the nitrogen in the drainage water. So we have to figure out which is which. So stack management practices may be important here, how we can manage these systems so that we lose the least amount. And there'll be trade-offs here as to what we can do and how we can minimize those losses. The winter Camellina relay system does show some promise. If you look at both the Camellina oilseed and the soybean yields, it does show some promise, but we have to optimize that. Do we get rid of the fall nitrogen application? Probably we should. Do we plant different types of row spacing so that there's not so much shading on those soybeans and maybe not so much competition for water? We don't know that. So that's something else we'd have to look at. And then the bottom line is, as always, weather effects outweigh anything that we can do out there. And since we can't predict the weather in any given year, that's a tough thing to do. The future then, what are we doing in the future? The common experiment is going to continue. I know there's funding for there's funding for the 18 sites from the USDA. It's in the budget for this next year. They're trying to get increase in budget for FY23, which starts in October, to fully fund some of the sites which aren't fully funded and hire more people, especially like data managers and other folks to help us out. So I know this is going to continue, and we'll continue our experiment as well. It's written into some 5-year plans, one of them that just is beginning now. So it's going to go at least 5 more years. Whether the treatments all remain the same, I don't know, but some of them certainly will. There are treatment changes that are possible. And I mentioned the strip till, because there's a field site up north of here in Williams, Iowa, where there's a comparison of a basic practice like we're doing and a strip till system. We don't have a strip till system in our plots at the Kelly farm. So that would be something that would be quite useful. The much larger area of these field trials means that you can do better measurements, like eddy covariance measurements, and look at carbon fluxes and such. And so we could compare some of the measurements in that field with what we're getting and see if what we're getting is true compared to a much larger area. Let's see. So the change in the nitrogen management. Yeah, we probably will do that. Maybe even replace Camellina. Maybe we'll try another crop, a cereal crop of some sort. We're really trying to get another crop growing in the system. And it seemed like this relay cropping system would be a good way to go if we can make enough money doing it. And then some data mining. So we've got piles and piles of data that we haven't analyzed, that I haven't looked at, that the other folks haven't in our group haven't looked at. We've got 6 years of data now we can mine. So there is some of that going on. Some of it you've seen already because I presented some of the yield data and some of the other things from all 6 years, but we've got a lot more to look at. And the other treatments. And then there's also organic systems. We have a whole set of plots at the agronomy farm, the organic water quality experiment that's been going on. We have not really compared what that system looks like as far as nitrogen cycling, carbon cycling, all of that with what we're doing at the LTAR site at the Kelly farm. So that's something else that we could do. And let's see, is that it? Yes. So thank you for your attention. Oh, if anybody's really interested, I have some of these handouts here on the LTAR system. This is about 2 years old, but you can pick one up because it talks about all the systems that are involved and what sort of things we're doing. So I brought a few of those over here. So comments, questions? Yes.
So I assume you had a block design for your plots?
Yes.
Do you have the opportunity to compare some treatments that may be on the Clarion versus the Canasteo?
That could be possible. I'd have to look. So the blocking at this site is a little different. It's not straightforward like you would think, because it was blocked on an EM survey that was done when, Kent, 2099? Okay, so an EM survey was done to look at the drainage out there because this was originally just a drainage trial. And so they blocked it on potential drainage at the site. So it's not just a straight block design like you would expect, but I think that would be possible to do something like that.
Sure. Well, beating on the drainage kind of hits on what I was curious about. It seems like the clearing areas would be shedding water, where the Canasteo would be receiving water from upper areas. So I think the water balances could be potentially interesting to look how that might affect some results.
That could be. I had a slide, I didn't put it in here, but the water, there seems to be, there's more water moving toward the north end, which would be correct, you know, because the Clarion's on the south end and the Nicolette's more on the south end. So it's moving toward the northeast corner. I've got a shot after we had a heavy rain out there of water about, 10 centimeters or less in the cornfield with the corn standing there and there's a flood of water there. It was about harvest time. So, but it did drain that area. So that's certainly something that could be done. Yeah. Any other questions, comments?
Okay, there's one that came in online. How tricky is the Camellina timing with harvest versus possible effect on soybeans and shading? Is that tough to manage?
Yes, is the easy answer. So yeah, I think the first year we did this, some of the soybeans, shall we say, got a haircut because you really have to watch the height that you're harvesting that Camellina. And if you get too low or the soil, that's the issue too, is if you plant the soybeans too soon and they grow too much, then you're leaving Camellina in the field. You're leaving the seed yield in the field, because if you get down lower, you're going to cut the tops off the beans, and you don't want to do that. So there's a lot of trickiness to that, to getting the Camellina and the soybean crop to grow to where they can both be harvested well, and you don't harm one versus the other and lose yield. Yes.
Got a comment from Anna Radke saying regarding the surface flow, we have seen some drainage data that suggests that's happening too, higher flows than precipitation. And then she also says that she's not volunteering to work on that.
Thank you, Anna.
And we have Dan Olk online who has a question. So Dan, if you want to go ahead and unmute and ask John directly.
You might have to repeat that, John, because the microphone's up there. Just say, Dan, if you have your question, just.
Oh, yeah.
Oh, there we go.
Is it my turn?
Yes, go ahead, please.
Yeah, sorry, my internet connection got shaky. John, I was real interested by your finding that one of your crop treatments reduced nitrogen losses to drainage in the drainage water, and a different treatment reduced the nitrous oxide emissions. Now, both of those losses primarily involve nitrogen as nitrate, correct?
Mm-hmm.
So why would one treatment affect nitrate loss through drainage and another one affecting nitrate loss through nitrous oxide emissions? Is it more the timing of the nitrate availability during the growing season? The timing is affected by those two treatments?
Wow, that's a good question. I don't know that I can answer that. I would think so, but I don't know that. I'd have to, yeah, I don't know off the top of my head. I can't answer that question.
Ryan's chiming in.
Oh, good.
Yeah, John, I'll chime in here.
Good.
Dan, I think you're partly right on that. We actually see something really similar when we're looking at unfertilized plots that are also out there. John didn't talk about them, but there's some plots that haven't received fertilizer since 2012. And those we see just the same nitrate losses from, but we don't see the nitrous oxide emission. And so what I think is happening is the overwintering, and yet the overwintering cover crops really making a big difference out there. And so that fallow period where things are leaching down into the soil and there's still mineralization, that source of nitrate of soil organic matter is really important in the leaching losses and not as important in the nitrous oxide as the in-season applications.
So it's more a matter of organic matter nitrate versus fertilizer derived nitrate. Is that correct?
Yeah, I think so. And I think the amounts that are there in the fertilizer derived nitrate when it's applied during the growing season versus the slow accumulation of what's being added to the drainage waters.
Okay, super. All right, Brian, thanks very much. Good
Yeah, thank you.
There's another question online, wondering, is Camellina with no-till possible? Maybe that would help with N2O losses.
No, the problem we have is that for weed control, when we plant the Camelina, we use Treflan and it has to be incorporated to work. And so, we've got the whole idea of what herbicides are out there as well. There is, and thank God, I think, there is no Roundup ready Camelina. So you can't count on that. And so we have to use Treflan, which has to be incorporated to work properly. So that's why, since we have to put that down with the Camelina, that's why we do the tillage out there.
I have a suggestion on cutting back the nitrogen in your Camellina. You could just use potassium sulfate rather than potassium nitrate. We're using ammonium sulfate. Yeah, so you can use potassium sulfate.
Yes. That would be good. Yeah, because elemental wouldn't work right away. Of course, the way the Camelina is growing, it probably wouldn't matter. It would, by the following spring, maybe we'd get some, even with the elemental, we get available sulfur. I don't know. Haven't tried that. But yeah, that's a good idea. Perhaps that's what we should do next time we plant Camelina, which will be next fall.
Your sulfur test didn't look that low, though. It didn't look like.
No, it didn't. It didn't. So we probably would be all right even without sulfur. So yeah, that's a possibility. But yeah, the potassium, since we seem to always be applying potassium essentially every year to these plots, that would be a good alternative instead of the ammonium sulfate, which we were using. Again, our thought when we started this was that, yeah, we need a little bit of nitrogen and some sulfur to get the Camelina off to a good start in the fall. So we're worried about winter kill, but it seems to do better. We tried to grow, I think, years ago, they were trying to grow canola out there, and we just had too much winter kill. And so, but now the climate's changing. And so, yeah, we'll see. I know one of my friends from Illinois has a farm by Ottawa, which is east of here off of I-80, and they're growing winter wheat and double crop soybeans now. So he said they started that a couple of years ago and they've had good luck with it. So it's like, I didn't know you could grow winter wheat this far north. I thought it'd be devastated by the weather, but nope, not anymore. So it's something else to think about when we come up with these aspirational systems.
It's hard when you started a treatment to switch kind of midstream.
That's the problem. Long-term research. It's like,
Yeah.
okay. But we've had that discussion already that I think Dan James and Tom Casper and probably Tom Mormon had a meeting with John Sawyer here years ago and said, okay, for our basic practice, what are we going to use? And he had some survey information, some other information for him and said, okay, this is what we're going to do for our basic practice, our business as usual. But now that's a long time ago now. So is that still the case? And how do you decide when you're going to change that practice? Like, is your basic practice no longer what people are actually doing? So you have to decide that in these trials. And I don't think we figured that out at this point. So, And, you know, we have all these work groups, and I think I mentioned we had 20 different work groups, at least in the LTAR network now. They're working on all kinds of things like that phosphorus budget. So some of those are morphing now into other groups or changing. I look at it like the Agronomy Society, American Society of Agronomy, with our communities. So when a community forms to work on something, eventually maybe they'll go away and another community will form because they've either solved the problem or, you know, it's not an issue anymore. So I think the work groups are kind of the same way.
Speaking of that phosphorus budget, could you pull that slide up? I wanted to ask you about one of the sites. It looked like there was a lot of phosphorus being added every year.
Let's see. There.
So there are 3 sites. That's positive, right? So surplus.
Yep.
Do you know where those sites are?
They are in Idaho, it looks like.
Okay.
And I would guess animal manure.
Yeah, that's what I was wondering, if that were manure sites.
Yeah. That's interesting. I'll look back at that paper again. There's a manuscript. I think it's gone to nutrient cycling and agroecosystems or something like that. One of those journals like that. So Mark Williams at the Soil Erosion Lab in West Lafayette was sort of lead author. He and Pauline Willike, who was at Purdue, but she's working somewhere else now. In any case, they put this all together and like gathered all the data from the sites and sort of did this meta-analysis to figure this out. And so I thought that was kind of a cool diagram to put in here because it shows where we are and also shows that we're fairly balanced. And I like to see that, whereas some of these other sites, like the ones in Idaho, are not. So we're doing well. Anything else?
Haven't had any other questions come from online. Anybody can ask?
We're good? Well, thank you so much. I
Thanks.
appreciate it.